Fungal infections destroy nearly 125 million tons of food crops each year, which could feed around 600 million people that have limited access to food security. One of the crops most affected by fungal pathogens is maize, which has a yearly productivity of over 1 trillion tons worldwide. Other labs have elucidated the process by which maize responds to fungal disease, but we are now in search of the pathways and pathology that allow certain fungi to infect maize plants. My name is Mackenzie Meikle, and I'm a third-year biology major, and my project has entailed performing a bioinformatic analysis of RNA-seq data in order to elucidate the molecular pathways of infection utilized by the necrotrophic fungus Fusarium graminearum and the biotrophic fungus Ustilago maydis. In this experiment, we studied both organisms in vivo and in vitro by culturing the bacteria in media and collecting a sample after it had infected a host maize silk. Both organisms were frozen and their total RNA was extracted. I then received the sequencing files and began my analysis, which is what my presentation will be focused on. The first step to a bioinformatic analysis is quality control, and this I've shown in figures 3, 5a, and 5b. Figure 3 depicts the mapping of Ustilago unmapped sequences, which are sequenced loci from our samples that did not match their corresponding reference, and we mapped these against the Fusarium data. Gray bars indicate that all the sequences are unmapped and there's a low likelihood of cross-contamination between the two organisms, since all reads were unmapped when checked against the other organism. Figures 5a and 5b indicate that all experimental and control samples behave similarly, which aids in confirming the statistical reproducibility and validity of downstream results. After these quality control steps, I was able to perform a differential expression analysis using the cufflinks package referenced, and more specifically, a program within it called cuffdiff, which quantifies the expression of the entire transcriptome between two experimental conditions. From this analysis, we can see the quantity and gene IDs of actively transcribed genes in the upper and lower 5% of the transcriptome, which are considered statistically significant genes. In figures 4 and 7, we can see that the infected tissue sample may have had less genes being significantly expressed, and we can see from figure 6 that the pathogens are actively infecting a plant have many downregulated genes in comparison to those existing without a host in culture. This has led us to believe that there are a select few genes that become active in the pathogenic fungi.